In this module, we discuss cleaning validation. Now, cleaning is undertaken by every industry. You do a lot of cleaning at home. Why is it so critical when it comes to pharmaceutical manufacturing? To put it in perspective, an average healthy adult male consumes around 1 kg of solids, around 4 liters of liquids, and breathes in and breathes out approximately 16 kgs of air every day. In contrast, when this male falls sick, a doctor prescribes a tablet, 250 mg, 2 times a day for 3 days. At the end of 3 days, the man has recovered. Within a week or 10 days, he is back to normal. That is the potency of pharmaceutical drugs. Now, where is 250 mg, 2 times a day, and where is the 1 kg of solids and 4 liters of liquids and 16 kgs of air? Now, he has got back to his feet because the prescription was right, the diagnosis was right, and things worked out for him. But what if it had not been diagnosed right? Or if the drug that was prescribed was right, but the drug that reached him was contaminated? GMP focuses on the core values of protecting and promoting safety, efficacy, quality in the drug products in commercial circulation. And one major area of concern is contamination. When product A appears in product B, the active in product A appears in product B, it is a very special case of cross-contamination. But contamination goes beyond cross-contamination to include just around everything. Now, if there is a contaminant present, it alters the identity of the drug, rendering the drug misbranded. If there is a contaminant in the drug, it vitiates the purity of the drug and renders it adulterated. So, the contaminant has done 2 things to place you at risk. One, it has rendered your product misbranded, you're liable for legal sanction. It has vitiated the purity and left your product adulterated, you're at risk for litigation. You place yourself at risk. Now this combined, being misbranded and adulterated, has compromised the safety, efficacy and quality of the drug. Now, contamination is something that every other industry also worries about. But in their case, a contaminant is defined as any substance or energy that adversely impacts their process. But in the case of pharmaceuticals, it goes beyond that. Of course, it could vitiate the process. It could ruin the process. But if it ruined the process and you're aware of it, it didn't leave your gate. But it did not ruin your process. Your batch was tested, the specifications were met, and the batch has gone into circulation and is now with the patient. What can it do? There are 4 possible scenarios. The first is a frivolous scenario, that it does nothing. The second is more serious. It could act synergistically. It likes what your active is doing and the contaminant gives a helping hand. Now on the face of it you might think that's a good idea. But just imagine, if the drug is a hypoglycemic drug, it is being prescribed to bring down the blood sugar from a certain level to a certain level. But because of the contaminant's synergistic effect, it has dropped way below to a point where the patient is now severely hypoglycemic and at risk. Or let's take a case of a hypotensive tablet. And it is supposed to bring down the patient's blood pressure from a certain level to a certain level. But this contaminant acts so vigorously, synergistically, to bring the blood pressure even lower to a point where the patient is now severely hypotensive and at risk, grave risk. Yet another effect it could have is that the contaminant acts antagonistically. Now, what's the risk over there? The drug, to have its effect or intended effect on the body, must bind at the target site to be effective. But here is a contaminant competing for the same site, and therefore it inhibits the effect of the drug. It does not permit it to bind at that target site. So what happens? 2 tablets 3 times a day and no effect. So the patient goes back to the physician, and the physician starts wondering whether he has diagnosed it right or whether the drug dosage was too low. So the patient is now at risk. The physician may unnecessarily increase the dose because he feels that perhaps he has prescribed too low a dose for therapeutic effect. Next, the contaminant could have an altogether different pharmacological effect in the body. For example, take a case where there is a hospital and one fine morning there's a long line of little children showing severe signs of allergy. Their eyes are watering, their breathing is affected, their temperatures are elevated, pulse rate is elevated, blood pressure is elevated, and naturally the children are in pain and bawling. Very soon it transpires that almost all the other hospitals in the neighborhood are also reporting the same event. A quick investigation quickly reveals that what is common to all these children is that they are all on the same multivitamin regime. And curiously, from the same brand, from the same batch. So, this is enough to alert the regulators who visit the manufacturing site. And what do they find? That the manufacturer had actually run a very strong systemic allergen in the previous batch and then taken on this multivitamin batch. Now, when a situation like this comes in, can you imagine what the fallout can be? In today's media-ruled world, you will be making headlines for all the wrong reasons. But this is a very strong possibility. But let me give you yet another possibility. You see, everybody doing cleaning validation makes a very common assumption that the equipment train from start to finish, bumper to bumper, is uniformly soiled. You see, you use your total area calculations. Why do you do that? You assume that if there is 100 centimeters here and the total is 100,000 centimeters, then 100 divided by 100,000 is the residue at that particular part. That is the assumption you're making. How valid is that? Next assumption you're making is that when it is worn, the residue worn off, it will be worn off uniformly. How valid is that assumption? Now you have some parts like your filler needles and your compression um, molds and punches. So at that particular point, there is a possibility in such machines that the entire residue of the previous batch that was there is transferred in one shot to one tablet. Because that was the residue in that particular cavity, and that was passed on to a tablet. Now it is being packed in your normal way and it is prescribed by some physician, and somebody takes it, and as soon as he has it, the patient is showing all kinds of abnormal symptoms. And assuming the person is not a sick person, a person is a normal person who is recovering perhaps and is having a multivitamin, and this happens to be a multivitamin, and suddenly he is showing symptoms. He goes to a physician and says, "Hey, listen, what's all this?" The physician is asking him questions about trying to diagnose. He is making no headway. Now you tell me, how seriously has the patient been placed at risk? First of all, he has been placed at risk with a contaminant that has gone in. Now the doctor, being at a loss to assess what could be the situation, is making a blind guess based on the symptoms that he's seeing, which could mean a totally wrong diagnosis, a totally wrong line of treatment. Now you place that patient at even further risk that he would be misdiagnosed and put on the wrong treatment. So these are the sort of concerns that one has when one is talking about contaminants in a pharmaceutical manufacturing environment. And cleaning validation becomes so very important because it could place patients at risk like this. Fine, then what is cleaning validation? Like any other validation, establishing with documented evidence that a specific cleaning process does the job it is intended to do and renders the equipment or line fit for reuse, meeting predetermined acceptance criteria and specifications. Now, talking about validation, when it comes to process validation, we know that the regulators have been talking about it for over 23 years. Now, why is it that cleaning validation is more recent? The focus, the emphasis on cleaning validation is barely a decade old as compared to 23 years of process validation. What could be the reason? One reason, of course, could be that any regulatory enforcement initiative is triggered by some adverse events in the field. So patients have got injured because of carelessness or inadequate procedures at manufacturer's level. But also there could be another reason. 30 years ago, it was not uncommon to have a physician prescribe 1000 milligrams 2 times a day for 3 days and so on and so forth. Batch sizes were usually in tons. These days, batch sizes are seldom more than 400 kgs. Very often it is 250 kgs or 100 kgs or even 50 kgs. If you were to go into the biotechnological field, a huge batch size would mean around 100 grams. For example, if you were the world's best manufacturer or the largest manufacturer of oxytocin, a peptide, you might find that the biggest batch you ever produce is less than 70 grams. Now, talking about 1000 milligrams and coming down to 50 milligrams and coming down to micrograms and then to nanograms and then to picograms, you can imagine the exponential rise in the potency of the drugs that are being introduced into the market. With that kind of potency floating around, if any one active gets into any other product, you can imagine what devastating effects it may have on the patient. This is the reason why there has been increasing concern on cleaning validation at the manufacturer's level. Now, who's asking for it? Naturally, the regulators. Now, if you have customers, then naturally no customer wants to place himself at risk, so he is asking for this. And finally, you yourself, could be initiating, focusing on proper cleaning for your own product quality, process quality. I'll give an example. If there is a fermentation batch and then after one batch is completed and the next batch is to be taken on, any leftover from the previous batch can contaminate that batch. His worry is not about the contaminated batch going into the market, because it will never go to the market, because the fermentation batch will fail, the fermentation process will fail. And then when you have a contaminated fermented batch, getting rid of that becomes yet another challenge. Sanitizing a contaminated fermentation processing environment becomes a very, very severe challenge. So to avoid such a situation, the manufacturer insists on very focused cleaning to ensure batch process quality batch after batch.